Signaling pathways are crucial in cell communication and, in particular, the regulation of the cell cycle. Without tight control of this process, organisms would struggle to ensure the right rate of cell growth, death, and replication. In this lesson on signal transduction, we will first begin by defining what signal transduction is. We will then examine the steps of the cell cycle because you can't really truly understand how it's regulated till you understand what encompasses the cell cycle. Then we will begin our exploration of the regulation of the cell cycle, which involves the structure and function of cyclin-dependent protein kinases, we call them CDKs, and their subunit cyclins. So once we understand their structure and how these complexes become activated, we can then examine the signaling pathway that actually controls the cell cycle. In biochemistry, we talk often about signal transduction, this communication within cells or between cells. Signal transduction is simply how cells take information and turn it into a chemical change or cause a chemical reaction. So this cellular change is very important to the life of a cell. Signal transduction is specific to each type of cell. It's also very sensitive. The molecules that bind to the receptors, such as ligands, they have a high affinity for receptors, so this participates in this specificity and sensitivity. And then there's this cooperation between the molecule and the receptor, which can cause large changes. It's called cooperativity. It doesn't take much, sometimes very small concentrations of ligands to affect the receptors and cause significant changes. So once this receptor or enzyme is activated by that particular molecule, it then triggers this activation, this cascade of other enzymes on down the way. And this is known as an enzyme cascade, which can amplify the message. Signal transduction occurs through four major types of receptors: G protein coupled receptors, ligand gated ion channels, and all sorts of enzyme linked receptors in the cellular membrane. And in the nucleus, we also find nuclear receptors. Now, in the cell cycle, we're going to focus mostly on kinases. So why do we need to take a look at the cell cycle? Well, without the cell cycle, life would be very different. We need the cell cycle to grow, to develop, to repair. I mean, without mitosis, an egg could get fertilized, but it would not split into 2 cells and then 4 cells and so on. In other words, the baby would not develop. Imagine all those holes that we have endured throughout the years from injuries, falling down and scraping our knee. Without the cell cycle, we would not make new cells to fill those spots in. Imagine how holy we would be. Sometimes DNA is damaged and it must be repaired before new cells are made. Otherwise, this mutation is just going to be passed on to the new cells. So there are controls and repair mechanisms in place to fix this. If the cell's not controlled, we get growth gone crazy. I know, sounds like a spring break slogan. And that result is often disease and cancer. Thus, a tightly regulated cell cycle is vital to the health of an organism. The cell cycle, in a nutshell, can be summed up very simply. Cells live and do their jobs, they make new cells. Cells live and do their jobs, they make new cells. Obviously, it's a little bit more complicated than that, but that's essentially what is happening. There are 4 basic parts to the cell cycle. We have G1, this is where we begin. So imagine you've got a brand new cell. It starts in G1, also known as gap 1. And this is where the cell is growing, it's doing its job, it's preparing for DNA synthesis. Now, during S, or DNA synthesis, we are replicating the DNA to ensure that there's enough DNA for 2 new nuclei during mitosis. And this is important because the new daughter cells need the same amount of DNA as the original cell. So humans have 46 chromosomes. Imagine if I gave you 46 socks and I said, could you please make me 2 piles, 46 socks, 46 socks? Impossible, right? But what if you could make copies of all those chromosomes? So you have 46 pairs of chromosomes. Now you can split them into two piles and get 46 and 46. And that's essentially what synthesis is for. So synthesis doubles the amount of DNA so that when we make new cells, we have plenty to go around. And then in G2 or gap 2, the cell again is just doing its job. It's growing, it about doubles in size, and it prepares for mitosis by making sure there's adequate proteins in the cell. Now, during mitosis, there's actually two things happening here. Number one, the genetic material, the chromosomes, are being separated into two nuclei. That is the first thing that has to happen. So in this diagram over here, we've got mitosis happening. So in prophase, the nucleus is still there, but the nuclear membrane begins to disintegrate and break down, and the chromosomes are free to move around the cell. And through a much more complicated process than this, the chromosomes line up in a single line, spindle fibers connect to those chromosomes, and then in anaphase, those chromosomes get pulled apart, and then by telophase, those chromosomes start going back into a new nucleus. The nuclear membrane reappears. So that's mitosis. Mitosis is just dividing up those chromosomes correctly amongst the two cells. Finally, at the very end, we have a separate process called cytokinesis. And cytokinesis is where the cell actually splits into two. Now, some cells have another phase, and you'll notice it over here, and that is G0. This is kind of like pause in the cell cycle. It might last for hours, days, it might last for the lifetime. See, cells like the hepatocytes and adipocytes, they have a very specialized function and form, and once they achieve that, they just go into G0 and stay there. Stem cells, on the other hand, well, they're not differentiated yet, and they can go through this process. So G0 is a really neat little pause mechanism, because if, for example, in the regulation of the cell cycle, it's determined that, there's an error in the DNA, the cell can go into G0 and fix those issues before the rest of the process goes through and we pass on that mutation. So this is one way that control of the cell cycle can happen and could hopefully prevent cancer. As we have seen, the cell cycle is very important, and it is regulated essentially through two things. One is called a cyclin-dependent protein kinase, or CDKs for short, cyclin-dependent kinases. But they don't work alone. They have to work along with something called a cyclin. And the cyclin, as you can see in this picture here, the cyclin and the CDK will join together to form a complex. And as a complex, they will work together to regulate the cell cycle. But just because they become a complex doesn't mean they're activated. They actually aren't. There are some steps that must take place. So because cyclin-dependent kinases regulate the cell cycle, well, CDKs and cyclins must be controlled and regulated. And how are they controlled and regulated? Well, the cyclin-dependent kinases are controlled through phosphorylation and dephosphorylation. We also have control of the synthesis and degradation of cyclins. And lastly, there are also inhibitors. So on the following slides, we're going to take a closer look at the structures of cyclins, cyclin-dependent kinases, how the complexes become activated and how they're controlled. And then lastly, we'll take a look at how do these different complexes work to regulate the cell cycle. Let's take a look at the cyclin first. Now, in this diagram here, you see two globules joined together. This top piece is the cyclin. This is the subunit that attaches to the cyclin-dependent kinase. And this has to happen in order for them to go on and become activated. Now, just because they're joined doesn't mean they can go do a job. They are still inactive. And in a few slides, we'll discuss how those actually go on to become activated. But first, let's just discuss cyclins for a moment. There are about 10 in the human body, and they are named with letters of the alphabet. So you will see later on cyclin A, cyclin B, and so on. They are named cyclin because they actually cycle during the cell cycle. In other words, they are synthesized, degraded, synthesized, degraded. Synthesis is controlled by growth factors and transcription factors. So growth factors and cytokines activate transcription factors, which are then responsible for transcribing genes that make proteins, including cyclins and CDKs. Cyclins are broken down when ubiquitin combines with the cyclin. And then that signals something else called a proteasome to actually come in and degrade the cyclin. And all of this is controlled by a very beautiful feedback loop. As the cyclin-dependent kinase concentration increases, cyclin joins so that it can go on to become activated and that complex can do its job. Well, once the complex is activated, it phosphorylates an enzyme called destruction box recognizing protein, or DBRP. Well, that enzyme breaks down cyclin. And so if the cyclin is being broken down, the CDK activity is going to decrease because remember, the two have to join together to become a complex. So if there's less cyclin, there's going to be less CDK activity. If there's less CDK activity, there's less phosphorylation of the DBRP protein going on. So it's just a really cool cycle that goes on and on and on to control how much cyclin and CDK activity we have going on during the cell cycle. Now let's take a look at the cyclin-dependent protein kinases, or the CDKs. Kinases, they have a job. Their job is to phosphorylate other enzymes or proteins. And as you can see in this diagram, that's exactly what this kinase is doing. ATP has those 3 phosphate groups. The kinase's job is to remove one of those phosphates from ATP and attach it to some sort of a molecule. And if you recall, this will help an unfavorable reaction occur. Most biochemical reactions are coupled reactions, where an exergonic reaction is coupled with an endergonic reaction. So that reaction has some energy to do what it needs to do. There are some significant structural components to a CDK that we need to be aware of. First, there is this T loop. We're going to talk about that pretty significantly because it often blocks the binding site for a substrate. We're also going to note over here this TYR15, this tyrosine 15 residue. When it is phosphorylated, it blocks the ATP binding site, and we're going to see how that is rectified in just a moment. Now on this diagram, you don't see it, but you'll see it in another picture coming up. There is a threonine 160 residue in the loop, and it plays a role in helping to activate the whole cyclin CDK complex. So again, you need to understand a little bit about the structure of cyclins and CDKs to understand how they can do their jobs. Recall that the cyclin-dependent kinases act as a unit with cyclin. So once they attach together, that's the first step in the activation process, but they're not activated yet. There actually has to be a phosphorylation and a dephosphorylation in order to become activated. So what you see in this diagram, and I like this diagram because it's just a little bit more clear. There is a phosphorylated tyrosine 15. That blocks the ATP binding site. Well, on the other side is the threonine 160 residue. When it becomes phosphorylated, then this one becomes dephosphorylated, and that finally allows ATP to bind. So once you have the cyclin and the CDK become a complex, you phosphorylate one thing, dephosphorylate another thing, then ATP combined and the whole thing becomes active. And this here is just a closer up view of that loop. And if you look very closely, you can see that the threonine 160 is in here somewhere. It just didn't show up in that other picture. Now that we understand how the CDK cyclin complex becomes activated, we can look at how they actually regulate the cell cycle. Now, there are 3 main checkpoints during the cell cycle. The first one is at the end of G1, right before synthesis. It is known as the cell growth checkpoint. It's also known as the restriction point. This is kind of like the point of no return. Once it goes past the restriction point, the cell has to go through the cycle. It's got to go all the way through to mitosis. Y'all, this is like the point of no return when you're on the way to the hospital to have a baby. You got to have the baby. It's coming out. There's no stopping it. So in order to proceed, the cell needs to be big enough and have made all the essential proteins needed to go on to synthesis. Moreover, there is a check at this point to ensure that the DNA is not damaged. In G2, there is a DNA synthesis checkpoint, and that's just a point where it ensures the DNA has been replicated properly and the cell has grown big enough to go through mitosis. The last checkpoint is at the end of metaphase in mitosis. It's also known as the spindle checkpoint. And basically, at this checkpoint, it just ensures that all of the sister chromatids are properly attached to the spindle fibers so that they can be correctly separated into new nuclei. Now, what we're going to take a look at next is all the different cyclin CDK complexes that regulate the cell cycle. We're going to see that in G1, we have cyclin D with CDK4/6, and that actually works in coordination with, at the end of G1 and going into S, cyclin E with CDK2. In GAP2, we have cyclin A and CDK2. We also have cyclin A with CDK1. And then in metaphase, we have cyclin B and CDK1. So what is it these complexes do? We'll take a look here in just a moment. Now let's get into the nitty-gritty on how these cyclin CDK complexes regulate the cell cycle. The first one we're going to look at is cyclin D CDK4/6, and that works in conjunction with, as you'll see, cyclin E CDK2. So remember, there is this point in GAP1 where there's no turning back, that restriction point. And this is where the cyclin D CDK4/6 operates. It is going to determine whether or not the cell goes through with cell division. So what is going on here is kind of complicated. Let's see if we can make this make sense. So right here in this picture, there's this complex of two molecules. The blue is called E2F. It must be able to bind to DNA to transcribe a set of genes that is required to start synthesis. Remember what comes after GAP1, synthesis. Well, this PRB protein is the retinoblastoma protein. It is usually attached to the E2F. So in order for E2F to break free and go do its job, the PRB needs to be released. This is where cyclin D CDK4/6 comes in. The cyclin D CDK4/6 phosphorylates, as you can see down here in this picture, phosphorylates the PRB protein, which then releases the E2F, which can then go on and do its job. Interestingly, when that happens, the E2F will signal to express cyclin E and cyclin A and CDKs 1 and 2. And then those will go on and continue to phosphorylate the PRB so that E2F can go on and do its job. The cyclin E CDK2 is also what creates this replication bubble in synthesis. Now let's take a look at a couple of other complexes. Cyclin A CDK2 or complex cyclin A CDK1 both act in G2. Now, cyclin A CDK2 does regulate in the timing and release of cyclin B CDK1, but the mechanism is not thoroughly understood. But these cyclin A CDK complexes play a role in contractile fibers and cytokinesis. And if it weren't for the cyclin A CDK1 complex at the end of G2, mitosis would not begin. The cyclin B CDK1 is activated around the centrosomes during mitosis. Now, this is where the spindle fibers are organized and formed. This is also known as the MPF, or the maturation promoting factor. Some people call it the mitosis promoting factor, which starts mitosis. Cyclin B CDK1 is very important in the actual process of mitosis. First, the cyclin B CDK1 complex targets lamin, and this is a protein that comprises the nuclear membrane. When lamin becomes phosphorylated, the nuclear envelope breaks down. Cyclin B CDK1 also plays a role in spindle formation, and as you recall from mitosis, it's the spindle fibers that attach to the chromosomes that pull the chromosomes apart. The CDKs also target actin and myosin, which are the contractile fibers that cause cytokinesis. So we need to have these complexes in order for the cell cycle to occur.
So we've already seen how cyclin CDK complexes control the cell cycle. But what controls them? Now we've already looked at, for example, cyclins and how they are synthesized and degraded and broken down. So there is that component going on, and there are all sorts of inhibitory and regulatory mechanisms in place to ensure that the cell cycle goes off unhitched. But I want to look at one particular very important inhibitor. So let's go to the beginning of the cell cycle, gap 1. Remember, there's that restriction point, that point where if the cell goes past it, it's got to go through with the rest of the process. It's got to go through synthesis, G2, and mitosis. But what if the DNA is damaged? Yeah, we don't want that damaged DNA getting passed on to the new cells. So what happens is when there is an issue with the DNA, P53 unbinds from the DNA and it goes and it activates another molecule called P21. P21 can then bind to cyclin E CDK2, making it inactive. So let's just back up and review what happens again in G1, toward the end of G1 before synthesis. First, you have cyclin D CDK46, and its job is to phosphorylate this PRB protein. And when it becomes phosphorylated, it breaks free. It allows E2F to become active. E2F then goes on and transcribes some genes that are required for synthesis. Well, if you have damaged DNA, you certainly don't want this to happen. Now, we know cyclin D CDK46 phosphorylates PRB, but remember, that complex can only phosphorylate PRB to a point. Then this cyclin E CDK2, it continues the phosphorylation of PRB, so more of those break apart from E2F so E2F can go do its job. So CDK 4,6 with the cyclin D is important. It does do some phosphorylating, but really a lot of the phosphorylation occurs with this complex. So we need to inhibit this complex, and that's what happens. So when there is DNA damage, P53 becomes activated. It activates P21, which binds to this particular cyclin CDK complex, keeping it from doing its job. So if PRB stays hooked to E2F, E2F can't become active. It can't go on, transcribe some genes that are required to go on to synthesis. And this is how the cell can stop the process for a little while. Now, what happens from here? Well, the cell could go into G0. It can go ahead and fix this problem, this mutation, so that it doesn't get passed on to new cells. And sometimes if that can't be done, the cell will then go through a process called apoptosis, where the cell dies and becomes degraded. Pretty cool process as well, but that's for another video. So what have we looked at in this video? Let's take a quick review. First, we defined signal transduction. Just a really fancy way of saying cell communication. Cells need to communicate with each other, and there's also a lot of communication that occurs within a cell. We also looked at the steps of the cell cycle. There's gap 1, synthesis, gap 2, and finally mitosis, where we make new cells. We looked at the structure and function of cyclin-dependent kinases and cyclins. When a cyclin binds to a cyclin-dependent kinase, a CDK, it forms a complex. That complex then regulates the entire cell cycle. But recall that just the binding of the two subunits does not make for an active complex. There needs to be some phosphorylation and some dephosphorylation and some ATP binding for that complex to become activated. And then those complexes go on and play a very important role in controlling the cell cycle. We looked at the different regulation or checkpoints throughout the cell cycle. We looked at how these various complexes actually operate within the cell cycle to control things. Lastly, we looked at how they can be regulated. Now, we already looked at cyclins and CDKs and how they're synthesized and regulated through feedback loops, but we also looked at inhibitors. For example, P53. P53 will recognize when there is a problem with DNA, and it will then turn on P21, which can then go on and control a whole CDK cyclin complex. But in the end, we need the cell cycle. We need it for growth, repair, and so on. But we also don't want out of control growth. So if the cell cycle is not tightly regulated, we will have out of control cell growth. And this, of course, leads to disease like cancer and quite often death.